In this video, we will be taking a deeper look at one of the four modes of prokaryotic transcriptional regulation. Gene regulation can be defined as the mechanisms that act to induce or repress the expression of a gene. Gene expression can be controlled at different levels, but in prokaryotes, regulation is mainly at the level of transcription. In transcription, the regulator protein binds to the operon. The operon is a segment of DNA that contains a group of genes controlled by a single promoter. There are different regions on the operon, including the promoter region, operator region, and structural genes. In order for transcription to occur, RNA polymerase has to bind to the promoter region. The regulator protein binds to the operator region, so depending on the presence of a small molecule and if the protein is an activator or a repressor, transcription may or may not occur. You can think of this like a gymnastics skill. The gymnast is trying to perform a vault, but the coach keeps blocking the runway to tell her corrections, preventing her from performing the skill. If the gymnast makes it down the runway, she'll be able to perform the vault, stick the landing, and receive a medal. In genetics terms, if transcription is able to occur, the structural genes will transcribe into mRNA. mRNA will translate into proteins, and the proteins will be used for some biochemical pathway in the body. To make this a little easier to understand, we'll look at one of the four modes of regulation, negative inducible control. Negative control means that the regulatory protein is a repressor and it will bind DNA to prevent transcription. Inducible control means that transcription will be turned on when the small molecule binds to the regulatory protein. So in the presence of the small effector molecule, the regulatory protein will not block RNA polymerase from transcribing DNA. In the absence of the small effector molecule, the repressor will bind to DNA and block transcription. We can think of this like an episode of Tom and Jerry. Jerry is trying to get to a piece of cheese from his hole in the wall. If Tom is around, he'll stop Jerry from getting to the cheese. However, if Spike decides to help Jerry out, he'll be able to stop Tom from messing with Jerry. In this example, Jerry getting to the cheese represents transcription, the repressor is Tom, and the small molecule is Spike. With Spike, Jerry can get to the cheese, and without Spike, Tom will stop Jerry. And these are my references.